Hello, everyone, and welcome to today's webinar. This webinar is hosted by Marianne Liebert, a Sage company, and sponsored by Ncardia. Today's topic is advancing gene therapy potency testing with iPSC-based systems. Thank you so much for being here. I'm Uduak Thomas, Senior Editor at Marianne Liebert, a Sage company, and I'm delighted to be your host for today's webinar. Let's meet today's speakers. You'll be hearing from Dr. Stefan Braum, co-founder and chief technical officer at Ncardia and Cellistic. You'll also hear from Dr. Shushant Jain, Director, Discovery Technology at Ncardia, and Dr. Silke Schwengberg, Associate Director for Cardiac Disease at Ncardia. We'll get started with today's agenda in just a moment, but first, some background on the topic. Gene therapies have proved transformative for a wide range of disorders, with multiple delivery platforms under active development. Recent FDA guidance emphasizes the importance of robust disease-relevant models to establish meaningful potency assays and ensure consistent product quality and clinical relevance. Induced pluripotent stem cell-derived systems provide a powerful solution to model physiologically relevant human cell types to bridge the gap between preclinical studies and patient outcomes. These systems improve the translational value of potency testing across viral and non-viral delivery systems, and they are well suited for assessing vector transduction, gene expression, and functional rescue within target cells. So in this webinar, our speakers will discuss strategies for implementing iPSC-derived assays in gene therapy development pipelines, with a focus on assay design, quantifiable potency metrics, and considerations for regulatory alignment. During the webinar, you will learn strategies for implementing iPSC-derived assays in gene therapy development pipelines, with a focus on assay design, quantifiable potency metrics, and considerations for regulatory alignment. You'll also hear case studies that apply these systems to mechanism of action studies, lot release testing, and comparability assessments. And you'll learn how to enhance the reliability, scalability, and compliance of potency testing across diverse gene therapy platforms using iPSC technology. Before we begin the presentation, I did want to let you know that at any time in the next hour, if you have a question for our panelists, you can enter it in the Ask a Question panel on your screen. All right, let's get started.
Thank you for the introduction and welcome to the Ncardia webinar about iPSC solutions for gene therapy. So I will be talking a little bit about the introduction of gene therapy and iPSC technology. Then we'll hand off to my colleague Silke, who will be talking about applications in cardiac disease modeling. And then Shui will continue to talk about AAV and CNS disease modeling and close off the webinar. So as a start, so there are different gene therapy strategies. Roughly speaking, on the left, we have ex vivo, more gene-modified cell therapies, and those really rely on tissue extraction, ex vivo gene modification. And so they typically then rely on cell-based assays to characterize them before they get transplanted back into the patient. On the right, which is the focus of this webinar, we will be looking at in vivo approaches, so relying on non-viral vectors such as polymers and lipid nanoparticles, and viral vectors such as AAVs, adenoviruses, and lentiviruses. And here the analytical strategies are a little bit more complex, because essentially what you're doing is you're having a delivery vehicle carrying a genetic payload or maybe a protein payload that is injected into a patient. And that has to find its way in the body, transduce or transfect those cells, which then have to change the disease phenotype, hopefully correct the disease phenotype. And as a result, the testing strategies are relatively complex. So what we're zooming into here is such a gene therapy test strategy for a viral vector. On top, we see some raw materials and cell banks. On the bottom, some plasmids. Plasmids are transfected into the cell banks, typically in a bioreactor. And the bioreactor produces viral vector, which is purified as drug substance, which is subsequently formulated to create a drug product. As you would expect, there are a number of identity and biosafety assays throughout the whole journey. And then where it gets really interesting is when it gets to potency. So how to measure that you actually have a viral factor that has the right potency, that does the right thing in vivo. So potency is something that is quite heavily regulated. So we see some definitions on the left. So FDA very clearly made sure that the therapeutic developer understand what they mean. EMA did the same, and regulators across the globe are doing the same, as well as the ICH, which worked on harmonization and basically clarifies to a therapeutic developer what they mean here. And so essentially what you see is a journey from preclinical to early phase clinical to commercial, where in early research therapeutic developers are really trying to understand what the mechanism of action is and how that mechanism of action links to the biological relevance and potency markers. So here it's really the goal to develop the therapeutic. There is no real regulation yet. Then in early clinical, obviously what the therapeutic developer is starting to look at is his IND submission. And in that IND submission, a therapeutic developer needs to have a description of the potency assay or the plan to develop one. There should be a demonstration that each lot released for clinical use meets a potency specification. And typically what you see is qualified, not fully validated. And then obviously what you would expect in later phases, clinical, so pivotal, commercial, you're going from qualification to validation. So you need to have defined acceptance criteria, and the regulator really expect that your potency assay is now consistent, quantitative, and stability indicating. So the bar really gets up very, very quickly, which is in many cases a real hurdle in these therapies. So then zooming in a little bit, pre-IND, we see things that are pretty standard, like infectious titer, capsid titer. Then early phase clinical, it's, as we said, surrogate. So it might be a messenger RNA, it might be a protein. And then where it gets really interesting is in later phases where it's now required to have this functional assay that measures the real potency of that cell therapy. And so now I'm going to make maybe a somewhat unexpected comparison to phenotypic drug discovery, because fundamentally we believe that there is a big overlap here. So this is a paper from Pfizer scientists from over a decade ago, and what they are talking about are three things that are important: a system, which is basically your in this case your cell-based system, a stimuli that triggers the disease phenotype, and a readout that is a relevant readout. And so, to not dwell on this slide too long, I'm going to shift completely to the right to high relevance. So what is described in this paper as a system with high relevance is something with a very clear link to a disease phenotype. So for example, an iPSC-derived cell with a patient mutation inside the genome of that cell, or maybe a patient-derived primary cell. That system can be as physiological as possible. So for example, 3D, for example, co-culture, or it might even have some mechanical stimulation. Then from the stimulus side, what you ideally would like to have is no exogenous stimuli. It's meaning that the disease phenotype is fully embedded into the cell. But the reality, and Shui and Silke will talk about that further, is that this is sometimes highly challenging, because obviously not every disease is manifested early on in life. Sometimes it actually literally takes decades before it gets manifested in a patient. So therefore, in a cell-based model, you actually probably have to stimulate. And then finally, you would like to have a functional manifestation of that disease phenotype in that cell. And so, for example, you can think about muscle contraction or endocrine function, secretion, energy generation. So something that is really fundamentally underlying the disease phenotype, so that you measure something that is as relevant as possible instead of the surrogates that we have been talking about earlier. shifting gears a little bit to Ncardia. So this is what we're doing, as you might have expected. We believe that iPSC are actually a great solution, and Shui and Silke will talk about that more, because fundamentally at first it's an indefinite source of patient-derived cells, and it can also help you to generate cells from tissues that are largely inaccessible. So cardiac and CNS are good examples. It will be very, very difficult to have a cell supply from a specific genetic mutation. In fact, you probably will not find that. And this is where iPSC kicks in. Furthermore, you can make functional readouts for efficacy, for safety, for mechanism of actions. And so in practice, what you will do is select the right donor or patient, create iPSC with Nobel Prize winning technology. So for example, you take a fibroblast, reprogram that all the way back to the iPSC state, and now you have a cell line that you can culture for a very long time. You can differentiate the cell to the cell type of interest, for example, the cardiomyocyte, for example, the neuron. And that is really the basis for your disease modeling, for studying that disease. And that work then forms the basis for potency assay development, validation, and ultimately transfer to a GMP facility. So in practice, how would this look like? So starting from left to right. So the first thing that you really want to do is to manufacture your cellular model, ideally in those bioreactors at scale. And you want to do that ideally as early as possible in preclinical development so that you can maximally benefit from that model. So you can early on start to study transfection efficiency, maybe transgene protein expression, maybe some toxicity, vector serotype, transduction efficiency. And then with that basis, you can start to study the disease phenotype and the ability of the candidate therapy to rescue the disease phenotype. And once you have that, hopefully preclinical, you have your basis for your potency assay validation and qualification, and doing this in a GMP certified facility to release your drug product. With that, we are wrapping up the introduction, and I'm going to hand over to Silke, who is going to dive in detail on the cardiac side.
Thank you, Stefan, for the introduction. Before diving into our cardiac disease model, I would like to highlight the importance of optimizing the delivery and safety of the AAVs to be used. In the case study example shown on this slide, we treated iPSC cardiomyocytes with 6 different serotypes of AAVs, each at 6 different MOIs. In figure A, you can see an exemplary image of the GFP fluorescence from 3 different of these AAVs. When calculating the transduction efficiency, all but AAV4 showed an MOI-dependent increase in transduction efficiency. When looking at the cytotoxicity, we observed some differences, with AAV serotype 1 and AAV serotype 5 showing a certain degree of cytotoxicity at the higher MOIs. Besides looking at a general transduction efficacy, we also checked if different versions of one AAV construct would lead to different target expression, both at mRNA and also at protein level. As you can see easily from figure A, both AAV showed an MOI-dependent increase of the target gene expression on the RNA level. However, these expression levels were way bigger with AAV model 2 compared to the AAV number 1. Interestingly, when looking at the quantification of the protein expression of the target delivered to the cells, we observed that both AAVs led to similar protein expression levels. So this data emphasizes how important it is to look both at the mRNA and the protein level expression of your AAVs. Over the next minutes, I will introduce one of our iPSC-based cardiac disease models that we have used here to test AAV therapeutics. But before showing the data from our first disease model, I would like to introduce our iPSC-derived ventricular cardiomyocytes to you. All our cardiomyocytes are produced in bioreactors. So you can see a little picture here in display number A. And as you can see, they express a very high level of the cardiac-specific marker troponin that you can see in this little flow cytometry image in figure number B. they show a very highly preserved sarcomere organization. And then if you look at the action potential displayed in this little figure here, you can see that this is indeed a very ventricular-like action potential. When you look at different batches of our cardiomyocytes produced, what we can show you is that both the cardiac troponin expression levels and also the viability of the cells are very consistent and very high, both at the time point of cryopreservation of the cells, but also up to 60 months post-storage in a post-thaw viability analysis. Lastly, if you look at the functional readout that we are using to qualify our batches of the cells produced, in the image in figure D, you can see the results from 8 different batches of our ventricular cardiomyocytes that have been tested in the microelectrode array. And you can see that the cells are fully functional and they have a very low inter- and intraval variability across the various batches. The first case study that I would like to introduce from the cardiomyocyte perspective is Friedreich's ataxia. Friedreich's ataxia is the most common hereditary ataxia with a prevalence of 1 in 50,000 people. It is mainly characterized by neurological and cardiac complications. Friedreich's ataxia is caused by mutations in the gene encoding the protein frataxin, which is essential for mitochondrial function. The deficiency of frataxin disrupts cellular energy production and leads to oxidative stress, contributing to the neurological and systemic symptoms associated with the disorder. In the vast majority of the patients, the mutant gene has 90 to 1,300 GAA trinucleotide repeat expansions in intron 1 of both alleles, causing epigenetic changes and formation of heterochromatin near the repeat. This then leads to the reduced transcription of the gene and in turn, of course, of the protein. Compared to healthy individuals, patients with Friedreich's ataxia might have only 5 to 35% of the normal frataxin protein levels. Besides the very dominant neurological symptoms of the disease, like impaired muscle coordination, slurred speech, and difficulties in walking, many of the patients with Friedreich's ataxia will develop cardiac-specific pathologies, and that will include impaired contraction, abnormal calcium handling, cytosolic iron accumulation, mitochondrial dysfunction, and then very often cardiac hypertrophy, which can actually be very severe and also life-threatening. At Ncardia, we first sourced iPSC cells from a patient, and then we optimized the differentiation protocol to be able to produce cardiomyocytes showing the genetic variancy of the disease. We then developed various assays that I will show you in the upcoming slides and functional characterized the disease cells and finally tested AAVs to see if we can turn around the production of the frataxin again. Once we successfully differentiated cardiomyocytes from the Friedreich's ataxia iPSC line, we first measured if the frataxin content of the cells is different between the wild-type control and the disease cell line. We did culture the cells in 2 different media. One is our basic cardiomyocyte medium, and the other was a medium enriched with iron, because iron accumulation is one of the changes that are observed in cardiomyocytes from the patients. As you can very easily see in the panel A, both with the normal medium and also with the iron supplemented medium, the cardiomyocytes have significantly reduced frataxin levels, which are displayed here as picogram per milliliter of the lysate. We were also checking of the cardiomyocytes from the patient iPSC showed an increase in ROS levels, which are indicative of the complications with the mitochondria. This was measured in a flow cytometry assay using a cell ROX dye. As you can see in panel B, we see a shift in both the fluorescence intensity and also the different populations that you can identify in this type of assay. Using a 4-gate strategy, we could clearly show that there is a shift from the low ROS expressing or showing cell population, which would be in population or gate number 1 and 2, up to the very highly ROS expressing parts of the population that are shown in gate number 4. And we do see a significantly upregulation in this later population, showing the very high ROS intensity in the cells. Last but not least, we were also looking for another functional marker of the cardiomyocyte, and for that we actually wanted to see if the cells will still contract normally. To identify any disruption in especially the calcium handling of the cells or any shift in the calcium handling of the cells, we were using a calcium transient assay for that. So the cardiomyocytes were cultured and then at a certain day were loaded with the calcium sensitive dye and then measured in the FDSS microcell instrument. As you can very easily see here in panel number A, which is displaying calcium transient traces of both the control and the disease iPSC cardiomyocyte preparation, there is a very significant and severe shift in some of the basic parameters of these calcium transients. So as you can easily see, both the duration of the calcium transient, so this is like how long it will take for one of these cycles to go from the bottom up to the top and then down to the bottom again, as well as also the decay time, which is actually like the slope from the top peak down to the bottom peak again, is significantly increased in the disease iPSC cardiomyocytes. And this is what is also observed in the patient and then will lead to some of the cardiac complications that are observed in the patients. Once we had the cardiomyocytes up and running and we were sure that they are actually displaying correctly the phenotype of the disease, we were then transducing these cells with an AAV that should lead to the production of the normal frataxin protein inside of the cardiomyocyte. In panel A, we again tested the frataxin level. So the left bar is the untransduced disease cell types, and the very right bar are the normal wild-type control cardiomyocyte. What you can easily see is that with these relatively high MOI, we could indeed see a highly overexpression of the frataxin protein in the cells. If you use the same MOI with an mock AAV, so not transporting the cargo that will lead to the production of the protein, you can see that there is no change towards the disease line in the protein content in these cells. Coming back to our functional assays, we could also confirm that in the calcium transient assay, we actually do see at a similar MOI that I'm showing you for the protein production, a reduction of this increase in the peak width duration of the calcium transient. So the calcium transient duration was going down after we treated the cells with these AAV. And then finally, also for the ROS production in the cells, we could see that these shift towards an upregulation in the highly ROS positive force population. So again, the 4 bar graphs are population 1 to 4. You can see that after the transduction with the AAV, the ROS production was going down and is now way more similar to the control iPSC cardiomyocytes. So in essence, we could demonstrate that not only we can make a successful iPSC-based disease model showing the phenotype of frataxin deficient or disease model phenotype of these iPSC cardiomyocytes, and then we were successful in also reverting this phenotype using an AAV that was delivering the healthy frataxin back into the cardiomyocytes. I will now hand over to my colleague Shushan Jain, who will introduce one of our neurological disease models to you.
Thank you for the introduction, Silke. So for this next part, I'd like to present the work that we've done in the ALS space. So really where we've developed a suite of assays to model ALS in a dish, and how we have then used those suite of assays to evaluate AAV efficacy in that model, and ultimately how the assays and the cell system can be transferred into a GMP environment. Just in brief, so ALS, it's a devastating disorder affecting individuals at quite a young age, and then causing a rapid death, normally within 2 to 5 years. It primarily affects nerve cells and really the motor neurons and the neuromuscular junctions, leading to muscle twitching, seizures, but also difficulty in speaking, swallowing, and eventually breathing. 90% of ALS cases have no known etiology, meaning they are sporadic. But in 10% of those cases, there is a known genetic component, and that has been very informative for the ALS researchers to be able to start finding effective therapies. And ultimately, in ALS, even though one protein is most commonly and most frequently associated with the disease. It's almost a spectrum disorder where ALS associated with a particular type of pathology is at one end, and then other diseases such as frontotemporal dementia, which can be associated with different genetic mutations, are at the other end. So as I've mentioned, ALS is can be caused by genetic disorders, but in virtually all the patients, there is a protein called TDP43, which is commonly found in the brains. And it's nicely depicted on the right-hand side of this slide on kind of the molecular deficits that are associated with the mutation of TDP43. So in a healthy state, TDP43 is located in the nucleus, where it has a role in controlling the splicing of a huge number of different transcripts, but one of them, one of the most important one of those is STAthmin2. And ultimately, by regulating splicing, it can then cause the neuron to behave normally. If, when TDP43 is mutated, it moves from the nucleus to the cytoplasm, where it becomes aggregated. This then leads to its own toxicity, but then also causes missplicing of genes like STAthmin2, to name one. And ultimately, all these molecular deficits collectively lead to electrophysiological dysfunction. And then that leads to neuronal dying, and then ultimately the phenotypes that we see within the patients. So at Ncardia, we have used human iPSC-based motor neurons in a monoculture or as in a co-culture to bring that relevance back, or bring more relevance to the model. And we've developed a suite of assays, both high throughput and functional, to begin assessing how one mutation of TDP43 leads to mislocalization, missplicing, and electrophysiological dysfunction. But then it also serves as a suite of assays to understand how your therapy, and in this instance, an AAV therapy, may be able to reverse these deficits. So what we have here is an example of the mislocalization assay and the TDP43 aggregation assay. As I mentioned earlier, TDP43 is commonly located in the nucleus, but when mutated, goes to the cytoplasm and then begins to aggregate. So in the images that we see here is in the top left image are just mutant motor neurons just treated with DMSO, and in the bottom left-hand graph are neurons treated with a stressor, causing reduction of nuclear TDP43 and an increase in the cytoplasmic. And this touches upon what Stefan mentioned earlier in his talk, where we want to have physiological model systems. We want to have those systems in place, but indeed in a lot of cell-based assays, it's needed to have a stressor to really bring out this disease phenotype. And then on the right, there is quantification of this mislocalization of TDP43 from the nucleus to the cytoplasm. And then in the bottom graph, this is also TDP43 quantification, aggregation quantification as well. So again, you can see both of these assays are very robust in being able to quantify both the mislocalization and its aggregation. So ultimately, we, as Silke also mentioned, we took a stepwise approach to understand how we can use this platform to evaluate the efficacy of a client AAV. So on the left here is motor neurons actually in a co-culture with astrocytes. They have been seeded, or they have been plated onto a multi-electrode array for assessment of electrophysiology. And what we can see is that if we transduce the cells 1 week after transduction, they are expressing low levels of the client therapy. But after 2 weeks of transduction, we have a very high percentage of neurons and astrocytes expressing the AAV, but they're also expressing quite high levels of the therapy as well. So once we'd actually optimized the delivery of the AAV into these co-culture systems, we then assessed as a first step to understand if the therapy was effective in being able to reverse TDP43 aggregation. So what we can see on the right-hand graph here is that with no therapy, there is substantial TDP43 aggregation in these co-culture systems. And when after we've transduced the AAV into the cultures, we can see an almost 75% reduction in TDP43 aggregation. And then a combination of using the MEA data, so the electrophysiological data, and looking at the molecular data of TDP43 aggregation allows us to simultaneously assess both AAV toxicity, but also the molecular rescue of reduction of TDP43 aggregation. So with that, I'd like to bring you to the last slide, where iPSC disease models are a powerful tool to be able to recapitulate key aspects of disease pathology. And this can be done both from a molecular perspective, but also more importantly, from a functional perspective. They can be used to understand how the AAV therapy can reverse the functional deficits of the disease phenotype, correlating with the intended therapeutic effect. And across a number of different diseases, we have developed iPSC-derived disease model platforms, which will not only allow us to, again, evaluate if the AAV is able to effectively rescue the disease phenotype, but it's platforms that are sufficiently robust that will enable us to test a new AAV batches for when you're further down your program. And I mean, I think it's a kind of an ethos at Ncardia where we really believe it's important to bring human biology and human functional relevant models and assays as early as possible into the drug development process, decreasing the risk of late-stage failure. And ultimately, although we've presented data across cardiac and neuro, the same approach can be applied to a number of different diseases where AAVs are being evaluated. So thank you very much.
That's all the time we have for today's webinar. Before we go, a note that this webinar will be available on demand shortly for repeat viewing or to share with your colleagues. Many thanks to our speakers for being here today and for a great presentation. I'd also like to thank today's sponsor, Ncardia, and the superb multimedia group at Sage for all of their support. Most of all, thanks to all of you for watching today. I'm Uduak Thomas, and from all of us here, thank you and goodbye for now.